Hello everyone, today I'll be presenting on microbial sensors in the food industry and I'll dive deeper into the principles and challenges of whole-cell microbial biosensors. In the next slide, you will see an article where we solely did our research on for this entire presentation. Now, imagine tiny superheroes working to keep our food safe. Those heroes are whole-cell microbial biosensors, or for short WCMBs. What are they? Simply put, they're living cells, either natural or genetically modified, that combine a biological recognition tool, like enzymes or receptors, with a transducer that converts their findings into clear, measurable signals. Think of them as built-in warning systems for food safety. So why are WCMBs a game changer? First, they're fast and efficient. They can detect harmful microorganisms, toxins, or chemical residues almost instantly, catching problems before they escalate. Second, they're portable and cost-effective. Unlike traditional lab testing, WCMBs work on-site and in real time, saving industries both time and money. Third, they're highly specific. Thanks to advances in genetic modification and nanotechnology, WCMBs can pinpoint exact contaminants or compounds with incredible precision. And the best part, they're versatile. Beyond food, they're used in environmental monitoring and even the medical industry. So what's the takeaway? WCMBs are revolutionizing food safety with their speed, accuracy, and affordability. They're working behind the scenes to make sure every bite we take is safe and high quality. As technology evolves, these biosensors will only get smarter, ensuring food safety stays a top priority. In short, WCMBs aren't just the future, they're already here, protecting us one test at a time.
Now talk about biosensor operating principle. A biosensor is an analytical device that combines a biological recognition element, or bioreceptor, with the transducer to produce a measurable signal proportional to concentration of a target molecule, called an analyte. In whole-cell microbial sensor, a genetic circuit is used, where the analyte interacts with specific recognition element, such as protein. This interaction triggers or represses the expression or reporter gene, part of the transducer element, under the control of specific promoter. Once the analyte detected, a visible signal is generated, indicating its presence. A well-designed genetic circuit is key, as it ensures the analyte is recognized first and the signal is activated efficiently. The first step in designing a whole-cell microbial sensor is identifying the target molecule or analyte, such as pathogens or chemicals, and selecting recognition element like enzyme, antibodies, or nucleic acid to ensure specificity. When the analyte binds to the recognition element, a signaling cascade activates a reporter gene, producing an output signal. Cases cell like Escherichia coli or Saccharomyces cerevisiae are widely used for biosensors due to their simplicity, rapid growth, and productivity. The recognition element ensures a specific binding to the analyte, which directly influences the biosensor's performance, including sensitivity and response time. Finally, the transducer element converts the recognition event into a detectable signal, often optical, such as fluorescent or colorimetric outputs. Green fluorescent protein is the most common reporter, but other proteins across the color spectrum are also used. Together, these components created efficient, specific, and measurable biosensors.
Now let's talk about how whole-cell microbial biosensors are the superheroes of the food world, saving us from sneaky villains like contaminants and pathogens. Imagine biosensors kicking out the toxic heavy metals like arsenite because no one wants that sprinkle on their salad, booting leftover antibiotics out of our fried chicken, and catching uninvited bacteria like Pseudomonas aeruginosa before they ruin your lunch. But it's not all bad, guys. Biosensors also help us find the good stuff like vitamins, B12 for that energy boost, amino acids, the building blocks of life, and organic acids, great for yogurt. Of course, being biosensors isn't easy. They're as fussy as divas on a world tour. Too hot? They're out. Too cold? Nope. Wrong pH? Say goodbye. Scientists solve this with clever tricks like encapsulation and freeze drying. Regulatory issues add another layer, as not everyone loves genetically modified organisms, but SIM-selves, safe and non-replicating, are the ethical solution. And then there's response time. Bacterial sensors say, "I'll be back in 20 minutes," while yeast sensors need a nap. Luckily, science is upgrading them to work faster. In short, biosensors are food MVPs, finding the good, fighting the bad, and keeping our tummies happy.
So what's next for biosensors? Well, imagine if your favorite gadget could not only survive the apocalypse, but also give you instant answers to your food safety concerns. Sounds like sci-fi, right? Well, the future of biosensors is closer than you think, and it's smarter, tougher, and way faster than ever before. Let's dive into how these little tech wonders are about to get a major upgrade. Imagine if your biosensor could handle a trip to the Sahara Desert or survive a sweaty hot yoga class. Well, thanks to spore-forming cells like Bacillus subtilis, that's actually becoming possible. These little guys are tough, they can survive extreme heat and even dry out without breaking a sweat. So now, we're talking about biosensors that could last up to a year no matter how tough the environment. They'll be ready to go whenever you need them. Now, we all know people can get a little freaked out about GMOs, right? But no worries, here comes SIM cells to save the day. These cells are completely GMO free, they don't replicate, and they only have the essential genes needed for biosensing. No risk of them causing any chaos in nature. It's just pure, ethical biosensing power that's safer for the planet and way more acceptable to everyone. Who doesn't love cool gadgets that actually work, right? Now imagine having a portable biosensor test strip you can just carry around in your pocket, ready to use whenever you need it. Thanks to hydrogels like alginate, these biosensors are protected and kept alive, even in harsh conditions. And the best part, these strips can be freeze-dried and rehydrated with water, making them perfect for on-the-go testing, especially in the food industry. Waiting for biosensors to give results? Not anymore. Scientists are speeding things up by tweaking genetic circuits and transcription factors to make responses faster. They're designing things like truncations and mutations to ensure biosensors deliver results in minutes, not hours. That means real-time food safety monitoring, quick, efficient, and exactly when we need it.
In conclusion, whole-cell microbial biosensors are vital in the food industry as they are able to identify analytes such as contaminants, toxins, and nutrients. They are also able to detect good contaminants including amino acids, vitamins, and organic acids, and bad contaminants such as pathogens, antibodies, and heavy metals in our food. They provide a wide range of advantages such as being cheaper and providing results faster and more effectively. The principles underlying these biosensors allow them to be higher in selectivity and sensitivity. However, they also face several limitations such as stability, regulatory restrictions, and response times. Despite this, advancements in nanotechnology and biosensors ensure that limitations of microbial biosensors will be overcome in the near future. With more research and development, microbial biosensors will become increasingly important and useful in the food industry to enhance food safety and quality. Thank you.